Could I ask folks to take their seats? We're going to get started in just a few minutes.
I can run the open public because we have that normal thing that we have to figure.
I never do at a workshop.
You don't?
I don't.
So that's an argument. So do you just want to call it?
I'll do that.
Okay.
Thanks.
Thank you.
I'll introduce you from that chair. Okay. Ask folks to take their seats and we'll get started. We're going to turn now to the issue of laboratory monitoring. And our first speaker is Dr. Lewis, also known as Vaughn to most of us, Kirchhoff, Professor of Internal Medicine and Infectious Disease at the University of Iowa, Carver College of Medicine. He's an expert in laboratory assay development for Chagas disease. He developed the RIPA assay for Chagas and is co-author of a large WHO comparative evaluation of serologic assays for Chagas disease. So Vaughn, thanks very much for being here today.
Oh, I want to thank the organizers of the meeting for inviting me. I only have 3 slides. And how do I move forward here?
I'm not an expert, so.
Oh, that's it. Okay, good. So I only have 3 slides and no pretty pictures or graphs or anything, but you'll understand in a minute. My plan or my charge was discuss the issues that should be considered by FDA or industry staff when they're considering the design of a trial to evaluate a new drug for Chagas disease or an old drug. And so what my 3 slides show is just really a summary or a list of the issues that I think they would have to think about as they work through this serious challenge, let's say. And a bunch of the things that are on the list here have been mentioned extensively by the earlier speakers, and I'll probably skip them, and so maybe I'll talk for just 8 or 10 minutes and then cede my time to Dr. Schiefman, who's going to talk about PCR. So if we can go to the first slide, general issues for evaluating drugs for Chagas disease. Evaluating drugs for Chagas disease is really a major challenge, as you can see from the meeting here today, but it's not uniquely so. I mean, this business of coming up with biomarkers and other indicators that are not clinical or patient-based indicators of efficacy, it has come out in HIV a lot and tuberculosis and many other diseases. In Chagas disease, as mentioned, there are no clinical related outcomes or no patient related or patient reported outcomes that would come up in a timely fashion that would help with likely help for an evaluation of the efficacy of a drug. In terms of the goals, my goal, and I think kind of everyone's goal, is parasitologic cure, to simply get rid of all the parasites that infect each person who acts as a host for this infection. But there's this question of whether suppression over a period of time is going to affect long-term outcomes, is kind of running in the background. In terms of what we can look at, parasitologic assays lack sensitivity, and specifically the old-time parasitology is with the insects, xenodiagnosis, where the insects are applied in a little cage on one's arm, and then you examine the bugs weeks later to see if they've gotten infected from the patient's blood. That's the old-time parasitology, and sort of in the middle is hemoculture. We take patients' blood or fractions of it and put it into an appropriate medium to see if the parasites will grow there. And they've essentially been replaced by PCR. They're much less, PCR is much less laborious and can be done in sizable numbers in a relatively short period of time. And then moving on to what I'm really charged with here is talk about serology. And this came up this morning in terms of how efficient our system is in the United States these days for screening the U.S. blood supply. And I think if you talk to the people, for example, Dr. Susan Stramer, who has been the head of serologic testing for our entire blood supply in the U.S., ask her about our Chagas disease assays, and she says they're excellent and they're really just as good as the assays that we have for other infectious diseases that do threaten our blood supply. So in a pre-treatment context for screening blood donors and for screening what I think and Dr. Maimandi and others think should be everybody who has any geographic risk for Chagas disease or maternal risk. All these people should be screened in the pre-treatment situation. And we have the serologic tools for doing that. And if you want to read about it, there was a comprehensive study in which was financed by WHO. It was published in 2009 in Transfusion. The first author was Otani, O-T-A-N-I, initials MM, and in that it describes their study, a study of, I think, 21 commercially available serologic assays that we use to test blood donor samples, positive and negative ones, from blood banks in 10 of the endemic countries. And there it shows that many, if not most of these assays are have sensitivities and specificities that are in the high 90s. And so those are the tests that are used throughout Latin America for screening the blood supply there, and I think they do a good job of it. Here in the United States, there's an ortho and an Abbott test that are used and for primary screening, essentially, of all donated blood, or all donors, I'll say all donors who are not known to be negative by previous testing. And then we do confirmatory testing, second stage testing, either with an Abbott test or with a test that I developed years ago called the Chagas RIPA. So we're in good shape for pre-treatment testing. In the context of post-treatment testing, it's much more difficult. And I'd summarize it by saying the variability and delay in the fall of anti-T-Cruzi antibody titers to whatever antigen is you pick after treatment make assessment of drug efficacy a difficult and prolonged process. And both Karen Byrne and Isabella Ribeiro showed slides of how titers fall, but they do so over years. And so in evaluating a drug, we really don't have the time to sit around for 4 or 5 or 6 years until it becomes clear that a drug is useful or not useful enough for us and won't be used. Okay. Yeah, just a couple of background issues that have been touched on by others. The question is, what group of infected persons would be the best ones to include in trials? And one of the issues is that I think the data show that people who have been infected shorter periods of time, like 2 years instead of 42 years, are more treatable or more curable. So if you want to power the test or you want to make it likely that a new drug or a new combination of drugs is shown to be effective, you want to do it on younger patients, just as a kind of a general rule. And then briefly, this business of reinfection was mentioned, reinfection after treatment. So someone comes in, is determined to be positive, gets in a treatment trial and gets treated, and then goes back to the context in which they got infected in the first place. And I don't, have strong recommendations on exactly how to do this now, but I'd say it's something that really needs to be paid attention to, because although there have been a lot of studies on the genetics of T. cruzi in recent years, I think at this point it's fair to say that we don't have a tool. For example, all the people who seem to be not cured, who might have gotten reinfected, we don't have a usable tool yet to look at the genetics of the parasites in those people to make absolutely sure that the parasites that they still carry are the old parasites rather than new parasites. So that's an issue just to think about. And again, in terms of selecting the group, as I mentioned, it's good in terms of serology to have a two-stage process. First, some kind of screening assay, and then followed by a more confirmatory assay. Another issue, it's very important, it seems obvious, but I think it merits mention, it's very important to avoid including subjects who are false positive in serologic assays. And historically, much less so now, but historically, false positive results in blood tests or serologic tests for Chagas disease have been a chronic problem. And so if you're working with an assay that's like, say, 99% specific, that means every 100 people you look at, you're going to have one person who appears to be positive for some reason unrelated to Chagas infection. And so if you work that kind of thing through the numbers of your study, in the end it could turn out to be important and bias your conclusions, your inferences in one way or another. So you certainly need to avoid that. And again, I don't have a shopping list here of a way to be 100% sure you can avoid the false positives. But one thing to think about is what range of titers in the screening assays do you want to include? And there's this kind of intuitive thinking that if a patient has just robust titers, as we say, they're pretty high, they are more convincingly true positive. And I think that's probably true to a certain degree, but not absolutely so. So whether or not to include that broad range or not is a question. Alternatively, you could just take, the top 25% in terms of reactivity in the screening assays, and then with either of these options, you could include PCR as a final confirmatory test. But then you're left with the question of whether are people who can consistently be demonstrated to be PCR positive. Presumably they have more circulating parasites. So either they're different in a biologic sense or in an immunologic sense, or the parasite strain they harbor is different. And there's no real way to sort that out, but either of those issues could conceivably affect the curability of their infection. So they're just issues to think about. Okay, moving on. A quick logistical issue. If you have a study with a large number of people, and if you adopt a perspective that you want to really be tight about doing your serological studies, and I think if the idea is to, instead of looking at the end of 3 years or 4 years or something like that, looking at the fall in titers in the months or a small number of years, if you're going to hang your hat on specific titers or levels of antibodies, I think it would be good practice to always run all the samples you have head to head in the same plot, and that would in the same plate, and that'll get rid of possible lot-to-lot variations. And it just, really appeals to me as one who's done a lot of serology to do that kind of thing. But the downside of that is if you've got 300 or 400 people in your study and you draw blood from them every 3 months, and you want to maybe get 10 or 12 aliquots with each one, because you don't want to take the same tube and freeze it and thaw it and freeze and thaw it, because that can affect titers. So all of a sudden you've got 10 freezers with 30,000 samples and four staff members to take care of it, and all your data up on the Microsoft cloud. And so it could certainly be a complication or a burden, but it's doable, and there's obviously computer programs to deal with that kind of data. Another issue is the long-term goal is to detect it's to detect after treatment, I would think, an early pattern of declining reactivity of antibodies or of some other biomarker that in the study or in a study has been shown that early fall has been shown to be indicative or highly predictive of parasitologic cure. I think that's the goal, and that's a real challenge, rather than, or in addition to with other biomarkers, waiting until they actually go negative. So moving on to targets, there are a number of things to look for. The ortho test, which is used to screen a good portion of the U.S. blood supply these days, and it's obviously approved by FDA. It's a size cut of lysates of T. cruzi epimastigotes grown in culture. So it has a broad and undefined mixture of antigen, everything from proteins to glycoproteins to glycolipids and who knows what else. So that's one approach. Another approach that many others and I have taken is mixtures of single or combined recombinant proteins. Wiener Laboratories in Brazil in Argentina has a 510K FDA approved, it's called a Wiener Chagas test recombinante that has, I think, 5 or 6 separately produced recombinant antigen in there, whereas the approach that I and my coworkers took was to do the molecular biology up front and make these strings of coding sequences of antigens that are from different places in the genome. So tests based on the latter is the Abbott PRISM Chagas assay, which is used also with the ortho test to screen the U.S. blood supply. Abbott Architect and the enzyme stripped assay that also has these recombinant antigens. So that's another approach, and there are tons of options in which proteins to include. And then there's whole parasites, and what we're particularly referring to here is there's an assay called complement-mediated lysate of whole parasites, living parasites, that identify a group of antibodies called lytic antibodies. And these lytic antibodies persist in people who have persistent infections. But when the infections are cured, then, as the supporters of this approach believe, that these lytic antibodies actually disappear. And there are people who say things like, it is widely accepted that assays that measure lytic antibodies are the best approach for assessing the effect of anti-T-Cruzi drugs. And so why don't we just, design a trial in which we do this complementary-mediated assay? And I think because it too belongs to old-time parasitology, you have to, it's very complicated. I've never done it, but I think you have to radiate mice, make them immunodeficient, and then you infect the mice with parasites from other mice, and then you eventually harvest blood from these mice, and I think by centrifugation separate the parasites from the mouse blood, and then run your assay. And it's complicated, and I think certainly in the United States from a regulatory point of view, I think it'd be difficult to convince the regulators that this is a good idea to do with this risk group 2 organism. I say that for years I've been chair of the Institutional Biosafety Committee at the University of Iowa, and I know my colleagues would raise their eyebrows thinking about all these potentially lethal parasites being carried around, between hoods into the animal room and stuff like that. So an option then is to let's say, clone or molecularly clone the protein essence of what the lytic antibodies bind to and measure those antibodies that way. And people are working on that. I mean, they've isolated what's called GP160, a glycoprotein from trypomastigotes, I assume, and using that as a target. And so there are papers out there that have used that natural molecule as a target to measure the level of lytic antibodies. And, I think more work needs to be done, and they're trying to clone it in a molecular sense as well. So then lastly, I'd say, well, what about all these different approaches, looking at both parasite and human biomarkers as indicators of infection status after treatment? And I'll just finish up with this. There are all sorts of things that people have been doing, with big science these days, including mass spectrometry, with MALDI-TOF and CELDI-TOF, and looking at expression of human biomarkers like APOA1 and FN1, and the list goes on and on. So if you're interested in this, this is an 18-page paper of which 3 of our panelists are co-authors that goes through all of this. And I think it's 2014, so it's recent information. So I'd say there's a lot going on, but the question is any of these approaches ready to go for a new trial? And I'll just read what they say authors say in the conclusion here of this review. They say there are several, and they reviewed the complement-mediated lysis business as well. So it's antibodies including all other biomarkers. Quote, there are several marker candidates which together may fulfill acceptable criteria to indicate the efficacy of trypanocidal treatment. Data from ongoing studies are considered essential to improve assessment of existing markers and to identify those for early follow-up of treated patients. So there's a lot going on there, but I think at this moment there's nothing that we can grab and order or bring over to the laboratory in the next month or two to help us address this problem. Thank you.
Thanks, Dr. Kirchhoff. We'll be now moving on to the issue of laboratory monitoring using PCR, and we've invited Dr. Alejandro Schickman to do that talk. He's head of the Laboratory of Molecular Biology of Chagas disease at the Research Institute of Genetic Engineering and Molecular Biology in Buenos Aires. He's worked in PCR assay development and exploration of biological markers of Chagas disease. He's the principal investigator for a WHO International Collaborative study conducted by expert PCR laboratories from 16 countries to evaluate the performance of PCR methods in the detection of Trypanosoma cruzi DNA by an external quality evaluation. So we're delighted to have you here today. Thanks.
Okay, thank you for the opportunity to share our experience and results. I'm going to talk on PCR standardization and validation issues in the context of using PCR as a surrogate marker for monitoring treatment efficacy. And you know that the next. Enter. Okay, what doesn't not working. No. See? No. Sorry, it was a technical problem. Okay. Okay, so the validation of PCR was recognized as one of the research priorities by the WHO, not only for case detection and characterization, but also as a test of cure. So since 2007, a series of international meetings were launched to search for a reliable test, PCR test, for monitoring treatment. Although PCR has been proposed for Chagas disease since the 90s, there was a high variability, a huge variability in sensitivity and specificity among the different techniques. So with the aid of PAHO and WHO, we started an international study to search for the most reliable PCR tests in all the laboratories with expertise for PCR in the world. For that, our task was to take into account the new knowledge about the genetic diversity of PCR, because one of the reasons to explain that PCR was so variable in the different regions could be due to the molecular targets used for PCR, which is related in turn to the genetic variability of the T cruzi. And nowadays, T cruzi is classified into 6 phylogenetic lineages or discrete typing units with different geographical distribution. So any molecular diagnosis should be analyzed in the context of this genetic variability, because the molecular sequences used for diagnosis have different copy numbers and different SNPs and mutations in the different strains of the parasite. So with this thought, we started to prepare a blind panel of samples, which was composed of one panel of DNA purified from strains of different DTUs, a panel of artificial blood samples that were spiked with different quantities of parasites, and a panel of real clinical samples from different parts of the world, and we sent these panels on a blinded basis to 29 laboratories in the world who wanted to participate in this task. CDC from Atlanta was one of those laboratories that participated in this. So these laboratories sent their results using their own PCR techniques, and we asked them to specify the DNA extraction methods, the molecular targets, the names and in composition of the primers and the rest of the protocol of the procedures. And we made a statistical analysis to search for the best methods in terms of specificity and sensitivity, asking them to have a sensitivity enough to detect a chronic Chagas disease patient, which had to be a very high sensitivity. And this experience was very useful to allow us to discard a lot of sequences of the parasites that nowadays we know that they are not useful for molecular diagnosis. Maybe they are useful for typing parasites of different lineages, but not for molecular diagnosis. The only two sequences that were useful for molecular diagnosis in terms of sensitivity and specificity were the kinetoplastic DNA, the minicircular sequence, and the satellite repetitive sequence of the nuclear genome, because they are the most repetitive genes per haploid genome and are present in all and very conserved and present in all the strains of the parasite. So among these sequences and the methods based on these sequences, the further choice was among the laboratories with good laboratory practices, because there were many laboratories that used the same sequences and same protocols, but the results were not the same. So a lot depended on the way of working of the laboratories. Another important conclusion of this study was that almost non-laboratory use an internal standards for the PCR. So they couldn't discriminate between a real negative result from a false negative results because of inhibition of loss of material during the procedure. So for that, we devised, after selecting four methods, we devised a plasmid to be loaded into the starting material, into the blood, and follow not only the amplification of T. cruzi genes, but also to follow the amplification of the internal standard. That is an imitation of the kits, of Roche kits, for example, for viral load in HIV and hepatitis C. And we distributed these plasmids to all the laboratories interested in performing PCR. So with this advantage of having not only a sequence specific for T. cruzi, but also for our internal amplification control, we set up, based on the best methods, a duplex PCR based on Chapman probes, with probes targeting T. cruzi satellite DNA or minicircular DNA, and the other probe targeting the internal amplification control. From this study, we could, okay, so right now, thanks. From this method, we wrote a standard operative procedure that is being used now in the clinical trials with new drugs. But we went on working with these methods, and in 2011, we made another international study where we transferred this technology to a lot of participants from all over the world who came to Argentina, and they were invited to bring their own clinical samples. That means samples from chronic and acute patients and patients infected with different lineages, to assess the sensitivity and specificity and the variability of parasitic load in only one laboratory, but the same operators using the same devices. And following the international guidelines of the Clinical Laboratory Standard Institute, we made all the experiments to have the operational parameters of the two duplex methods in terms of precision, limit of detection, limit of quantification, analytical sensitivity for the different strains, and also exclusivity for microorganisms that can be involved in the same samples of patients with Chagas disease, like as patients with T-Branchelli infection or patients with leishmaniasis. So provided the good sensitivity and specificity of these methods, we transfer them to all the population, and these are now available for everyone that are published to be used in research and in clinical trials. This is an example of the results, and here you can see the variability of parasitic loads measured by both duplex PCRs in clinical samples infected with different discrete typing units, and we can see that there is no statistical difference between the measurements using one or the other PCR method. Another important issue that we found out that to perform a good reliable PCR, we must have external control quality standards. So we started to provide all the laboratories that wanted to do PCR by blind panels made of different strains with different quantities of parasitic loads that they are composed of seronegative blood infected with culture parasites, with known quantities of culture parasites, and they have a blind code. They are sent to the laboratories, and we ask them to give us the results, and at the same time we perform our own experiment with the liquids of the same samples, and we see the concordance of the samples. And here you can see, for example, that for different laboratories, these are 4 panels, which is we prepare panels for 1 year of work. They have to perform the PCR each 3 months. And you can see that although there is difference between the values of PCR using different strains, which is natural because different strains have got different copy numbers of the targets, there is no statistical significant difference between the mean parasitic loads from one panel to the other panel. That means that the blood samples that are taken into a stabilizing agent, like guanidine, for example, they are very stable, at least for a year after getting them and transport. This is important because in some clinical trials that are made in endemic regions, you have a time before the collection of the sample, the transport to the molecular laboratory, and the procedure itself. For example, you can see here different panels of 1, 10, and 100 parasites per ml of blood, and you see that the mean values of the parasitic load are homogeneous, and the differences between the median and the percentiles is due to the multiple strains used in this, but we know that patients are infected by multiple strains of different tissues, but so it's important not to expect very homogeneous results because of this variability. And another issue of this is that the laboratories that participated in this external control, they were able to look at the results and to improve them. For example, this is an example that for the samples with less number of parasites, which were close to the limit of detection of the method, they had negative results, and then after changing the real-time device, for example, and making some changes, they could detect them. So it's an important means while they are working to improve their results. And at last, I wanted to talk about the application of these PCRs in field studies and clinical trials. One of the works we have been done in collaboration with MSF at NDI was to, in an attempt to improve sensitivity, clinical sensitivity of the test in chronic patients. In particular, in patients from Bolivia, the parasitic loads are very, very low. These chronic patients have parasitic loads that are about the limit of detection of the PCR. So the way to improve their sensitivity was to optimize sampling, and we tested 10 milliliters or 5 milliliters of blood with a stabilizing agent taken in the same day, and another one taken 7 days after, because you know that the natural history of the chronic infection, you have peaks and valleys of parasitemia. So it depends on the time you get the sample, you can have higher or lower parasitic loads. And here we demonstrated that, of course, the use of serial samples, the use of 3 samples, improved the clinical sensitivity of the PCR from 87% to 91%. So, for example, for the E1224 trial of DNDi, we chose this strategy because the patients in Cochabamba had very, very low parasitic loads. This is different. This must be studied by a pilot study. When one chooses a population, it is good to make a pilot study of the parasitic loads in the region. For example, in Colombia, patients have high parasitic loads. So the strategy may depend on the region where one is going to do the assay. So in this study, we saw that there was no difference between 5 and 10 milliliters, or in blood taken the same day or 7 days later, but there was a difference between taking a positive result for a patient, taking 1 sample, 2 samples, or 3 samples. These are the results using the same kind of PCR that have an external control from our laboratory, the Chagas study. And you can see that, as Isabella Ribeiro showed you before, that the usefulness in the short term of the PCR to show treatment failure. So this is real a main usefulness of PCR, because in the short term, at least you can say that the drug is not working. If you have a negative PCR in a chronic patient, you cannot say that the patient is cured, because you are always taking a blood sample, and so you are evaluating the parasitic load of the bloodstream parasites, but not the ones which are in the tissues, in the target organs, like, I don't know, the digestive tissues or heart tissues. But if the result is positive, you can say that the treatment is failing. There's another example that's already shown by Isabella, looking at the differences using this PCR between placebo, Benzene, and the different arms of E1224, so I'm not going to insist in this. And this shows the fluctuation, the variation of the parasitic loads of the E1224 study, and you can see that the placebo arm, there was a persistent parasitic load. The arm with Benzene decreases in parasitic load down to undetectable levels and persisted undetectable, and the arms with E1224, depending on the dose, they started to relapse after treatment. So the follow-up with PCR is also a good tool to monitor. Also, from this study, we saw that there was increased hazard of relapse with treatment group and higher quantitative PCR baseline. So the parasitic load also can be an indicator of the prognosis of the treatment. At least in this study, the patients who had higher parasitic loads at baseline had higher hazard to relapse after treatment. Okay, that's all. Thank you.
Thanks very much. Appreciate it. That was a great talk. I'm going to rearrange the agenda a little bit and take open public comments at this point so that they can be considered by the panel during the second part of the panel discussion. We have one request for formal open public comment, and that's Mr. Frank Sazanowski representing Mundo Sano.
Thank you, Dr. Farley. Frank Sazanowski from Hyman Phelps McNamara, representing Mundo Sano, Spanish word meaning healthy world, through our work with the Chemo Pharmaceutical Group. Mundo Sano is a nonprofit foundation that was started in Argentina in 1993. Its sole mission when it began was to combat Chagas. It has expanded its mission to include all neglected tropical diseases since then. And it has worked with many international coalitions, and in that work has worked alongside the Gates Foundation, the World Bank, WHO, PAHO, and others. Mundo Sano has two requests for FDA to consider today. First is to consider the use of Subpart H for Chagas therapies, and the second, to consider recognizing Chagas as a neglected tropical disease. Mundo Sano believes that we have heard here today many reasons why a therapy for Chagas should be considered under FDA's Subpart H program. So what is Subpart H? It's also known as accelerated approval or fast track, and FDA created it back in the 1980s to respond to the AIDS crisis. It allows the FDA to approve a therapy based on, quote, a surrogate that's reasonably likely to predict, close quote, ultimate clinical benefit. But only if the disease is serious, and only if there's an unmet medical need. I think today we've heard that undoubtedly Chagas is a serious disease and that it is indeed an unmet medical need where there's no approved therapy here in the U.S. So why would a therapy for Chagas be based on a surrogate and not on actual clinical benefit? Well, Mundo Sano thinks that because with Chagas, a study to prove the ultimate clinical benefit, improved survival, would likely take a decade or longer to run. A drug therapy could be approved on a surrogate much sooner and allow patients to access treatment through the normal health delivery system rather than through the compassionate use system. Today, the CDC runs the only such program for Chagas in the U.S. But to get access, a newly diagnosed patient would need to be in the care of a physician who knows Chagas, knows of the CDC program, is willing to invest the time and effort to personally apply for such access. Also, there's a well-known phenomenon that when the FDA approves the first real drug for a disease, the awareness of the disease among physicians, and therefore the number of patients diagnosed, expands, sometimes dramatically. Recall when Tagamet was first approved for ulcers, when Prozac was first approved for depression, when Betaseron was first approved for multiple sclerosis. The same could be true with the first therapy FDA approves for Chagas. So how could FDA apply Subpart H to Chagas? Well, FDA could rely upon the findings on a surrogate, such as the quantitative PCR test that Dr. Shipman just described in the recently completed Dr. Molina study out of Barcelona. Thank you. And the FDA could look at that kind of surrogate finding as the basis for accelerated approval, with then a post-approval or phase 4 study that would confirm the clinical benefit, such as the reduced incidence of sudden death, thromboembolism, and heart failure, which are the leading causes of death in Chagas heart disease, the most serious and frequent manifestation of chronic Chagas. This use of Subpart H would follow many FDA precedents. Since 2010 alone, And just in the anti-infective area, the areas that Dr. Cox, Dr. Farley, and Dr. Nambiar and her colleagues work in, just in the area of anti-infective since 2010, the FDA has approved many drugs for HIV and for hepatitis C based upon the results of blood tests for these infections rather than clinical benefit. In December 2012, FDA approved Serology to treat MDR-TB based on the absence of a pathogen in the sputum coughed up from the lungs. Mundo Sano asked FDA to consider applying Subpart H to Chagas therapies in order to accelerate patient access to a therapy for Chagas. And if that's done, it will likely result in expanded physician awareness of Chagas, and then that would lead to many new diagnoses of persons in this country infected and at risk. Mundo Sano's second request to FDA is to recognize Chagas as a neglected tropical disease. The original 2007 law that created the list of neglected tropical diseases failed to list Chagas. And this law also saddled the FDA with such a cumbersome rulemaking system for adding any new disease to the original list that FDA even found it difficult to add Ebola. So in December 2014, Congress and the President changed the law. They added Ebola and, importantly, gave FDA new authority to add diseases without being encumbered by rulemaking process. So on behalf of all those afflicted with Chagas in this country, both diagnosed and the many more now undiagnosed, Mundo Sano asked FDA to make Chagas the first disease that FDA adds to the official list of neglected tropical diseases under FDA's new December 2014 authority. So in sum, Mundo Sano asked of FDA both to use Subpart H for Chagas, to recognize Chagas as a neglected tropical disease, and on behalf of Mundo Sano, thank you. Gracias.
Thanks, Mr. Sosnowski. And just to clarify for the panel, I know that he did say that, but Subpart H that is being referred to is the accelerated approval pathway that Dr. Turner described in his talk. They are synonymous. That's the subpart that refers to accelerated approval as a type of approval. So we'll move on to the panel discussion, and I'll turn it over to Dr. Nambiar.
Thank you, John. So I think we'll move on. We have a fair number of questions that we wanted to cover in the second part of our panel discussion. Some of them we've already touched upon in the first session. So before we delve into the questions, I just want to see if any of the panel members had any lingering comments regarding either the populations or the acceptable control groups. If none, then we can just go into these sets of questions. Okay. So the first question we seek your input on are what are feasible and acceptable clinical trial designs? And in Dr. Turner's presentation, he gave you some options as to what are the different considerations for a trial to be an adequate and well-controlled trial to meet regulatory requirements. So would welcome thoughts from the panel members on what might be various trial designs. Sure, Dr.
Well, I think that, as we discussed before, I think that we have in the case of adults, I think a placebo concurrent control would be acceptable, depending on the timelines for follow-up. So a 6 to 12 month follow-up, I would say that is actually acceptable still. In the case of children, I don't think basically a concurrent placebo design not acceptable, and therefore we are discussing here either the option of actually having a dose comparison concurrent design or an active treatment concurrent design, or alternatively a historical control. So that, in terms of the different target populations, those are my views in terms of adequately and well controlled. I think that I should mention that for adults, both obviously the active therapy concurrent and the dose comparison concurrent designs would also be acceptable. That's my view.
I completely agree with the exposition of Isabella, and just to like to add that blinded, if it's possible, it depends of the kind of drug that are being tested.
Dr. Ribeiro, can I just ask you a clarifying question? So when you had suggested that dose response studies might be an option, and I certainly don't know enough about this field. And how do you sort of assess what might be potentially useful doses? I think here, historically, we've just used a dose or two and we've gone 60 days. How do you make an assessment of would 30 days be enough, would 14 days be enough, would 60 days be the right, and what kinds of doses? Do you base that on animal models, or do you have some other way of doing it?
That's a great question, because we've been dwelling with this at this point, and I think that there is no clear, we don't know yet what would be from the animal models, the duration in animal models that would reflect that clearly in the animal models, if you look at different durations, you have an increase in response. So you have, like, you test 5 days, 10 days, 2 weeks. 40 days, for example, you may see changes, but obviously this will depend on the drug and so on. So at this point, what we have evaluating in the case specifically of we are right now evaluating a design for a benznidazole trial. We're actually looking at 2 weeks, 4 weeks, and 8 weeks treatment duration. And we are actually following patients. So we have assessment of end of treatment response, and then 6 months and 12 months, looking at sustained response. I hope I answered your question.
So,
and we also do, obviously, now with the information that we have in terms of PK/PD and some of the data on quantitative PCR, through trying to model and actually try to develop to actually predict and having model predicted responses. So that's actually one of the outcomes that we're going to be looking, particularly for phase 2 in the future. We're considering.
So when you had your results from your azole trial, I think the Chagas azole trial, were you sort of surprised, or did you have enough priors before you went into the study sort of predicting what might be the outcome?
There was quite a bit of information that had been generated on posaconazole in acute and chronic Chagas models. In the case of E1224, we actually had studies also in murine model, mostly 20-day model with immunosuppression. And actually the response with azoles were excellent. But it looks like there is now some additional data that's actually showing that depending on the strain, they are translatable, that you might have translation. So, for example, there is now data from the Cell-Brenner model, the Cell-Brenner chronic model, that one could see that one would have failures with this. And so now we need to accumulate additional data to really support that this will, at least for as we evaluate additional compounds, that this will remain translatable. Translational.
To Dr. Sosastani and then Dr. Alchi, do you have a comment to make?
Really, there is no rationale for 60 days of treatment. During several years, we use 90 days of treatment. Afterwards, we use 60 days, but we can use 30 days of treatment. There are some publications about 30 days of treatment. Then we have to include 30 days of treatment in all of the designs for new drugs, because there are a lot of evidence that 30 days could be a good time for treatment. Maybe less, but at 30 days will be okay. Other thing is about doses. Nobody knows the doses that we need. We found that children need lower doses, that we are using overdosing for adults, for example, for benznidazole. We do not have enough information about nifurtimox, but we will generate new information in a few months about PK of nifurtimox.
Certainly help make the safety profile better as well. Certainly. Yes, Doctor.
Yes, I'd like to make a difference regarding the standard scheme with several questions that we have up to date to review regarding the old drugs. The reason for using 60 days up to date in most of cases, as in most of guidelines, is because the unique clinical trial during 19th was performed with 60 days. And in addition, as Jaime said, there are a lot of information with observational studies showing that 30 days prevent morbid mortality and reduce antibodies and become negative an important percentage of patients. Several comments regarding 60 versus 30, 5 milligram versus 2.5 are new questions to be evaluated.
Any other comments?
Obviously, we need pediatric formulations too. Now they know we have to say every time and in every place. Sorry, but we need pediatric formulation.
I don't think you'll have to convince me on that one point. So that's easy. So I suppose if there are no more comments on this question, we could move on to the second question. And again, we've certainly touched upon this already, and it came across in the two presentations we had from Dr. Kirchhoff and Dr. Schickman. So the question really at hand is, what would be an appropriate primary endpoint? And again, to consider whether a clinical outcome endpoint is feasible and what are the strengths and weaknesses for that, and certainly the timing of the assessment of this endpoint and how long patients would need to be followed up. So maybe we can take it a piece at a time. Probably have a discussion on what might be the endpoints, talk about both the surrogate endpoint based on a microbiologic criterion, as well as a clinical outcome endpoint, which I think for the most part, as we've heard, is difficult to do. And maybe after we have results of the benefit trial, it may be even harder to do. But would welcome the the panel's thoughts on that.
I will try to respond the second questions, because there are at least three very important questions. Regarding the clinical, with the primary, if a clinical endpoint is possible, I think that we discussed it a lot, that at this moment, maybe always is not possible to use. Regarding the question, when should treatment benefit be assessed? I think that is, there are, at least in this moment, a consensus that during 12 months after treatment is an excellent time. And certainly using PCR and serological tests, also looking for reduction. So it's important to consider to assess failure by PCR and success by reduction of antibody. Regarding the value of negative PCR, I think that would be very important to be clear that the important result of PCR is the positive result. Regarding the strength or weakness of serological tests or PCR, Personally, I think that the strength of serological tests is that it's more sensitive than PCR in general. And the weakness is that the after treatment reduces slowly. And the strong PCR is more specific after treatment. And the weakness is that negative result don't mean cure. It's a crucigram.
Yeah, I think that's clear. I didn't have any follow-up questions. Did you have a comment, Dr. Ben?
I think perhaps that, particularly in children, I think the statement that a negative PCR, I think a negative PCR, it should be considered as, for me, there is, I think it's cured. and in adult, I think that, and I think in adults, I think the issue of sensitivity of the PCR, it's more of an issue. But for example, particularly in children, PCR sensitivity is in the order of 100%. Please, Dr. Alcek and Alejandro, please correct me. But it's really with pretty good specificity too. So here it's where I wonder. But I think we were not. I just wanted to ask a question in terms of the first question of what primary endpoint would be appropriate for a clinical trial. To answer such a question, should we take into consideration for a clinical trial for marketing authorization, for efficacy demonstration? That's the continuation of that question.
Yeah, I think it's for purposes of being able to prove it.
So from my standpoint, I think it's interesting to think about, and I think we did briefly earlier, and I think in that sense, the presentation that we had before is that in one hand is that are we ready to say that PCR, for example, or serology are validated primary efficacy endpoints, and what we will need to consider PCR/reduction of titers across different age groups as a validated primary efficacy endpoint. Therefore, one would not need to demonstrate later on clinical benefits, right? The other one is actually, oh, I'm so sorry. I preempted the question, I apologize. And so the other one is in relation to, should it be considered more a surrogate marker that will need still subsequent demonstration of clinical benefit. But I didn't want to, but because I think today we are moving certainly in a position where I am of the position that indeed it should be considered for children response, no doubt, I think we are there. and in the case of adults, I think that particularly with the benefit trial results, I think the correlation between the validation of that outcomes will be there.
Dr. Kirker.
Yeah, I'd just like to comment on the serology business, what to do with serology. From the perspective of serology, PCR just looks really kind of simple and, one thing, and I realize there are complexities of the strains and things like that. But, having spent some time looking over a lot of these studies relating to serology, I would say, evidence that change in serology is seronegative or reduction in titers. So antibodies to what? I mean, in terms of recombinant antigens, there are just literally dozens of recombinant antigens that go back really to the late 80s. And then there are hybrid recombinant antigens that are combinations of these. And then there are native antigens. And then there are, like I said, size cuts of separated molecules from lysed parasites, and we're talking about, a substitute target for the lytic antibodies that is the protein core of the GP60 in recombinant form or isolating native GP60 from trypanomastogotes, and all of a sudden we got a list of 38 targets. So the question is how to sort that out, and one possibility would be to go back and shake everybody's freezers from trials that have been done before. We have specimens for treated patients. But the problem with that is, there are no temperature records that weren't collected in FDA-certified contexts. They've been frozen and thawed, undetermined number of times, and so it's a tough and very heterogeneous group to work with, so that's problematic. So at the other end, you could, in a sizable trial, just collect lots of specimens and lots of aliquots and take this panel of targets for antibodies and other biomarkers and just do it. But, it's going to take 4 years or 5 years to do. So I don't know what the answer is, but I just wanted to respond. Evidence that change in serology. Yeah, maybe the benefit trial. I don't know how much serology they have, but
Yes, I would like to take advantage of Louis' comment regarding that, making one more time the difference between the situation during clinical trial in comparison with point of care. Certainly, up to date, maybe for the point of care, certainly the PCR is the more practical to demonstrate in a short time a benefit of treatment, showing that don't have failure. And regarding clinical trial, you said during your presentation a very important topic regarding about maybe if a profile of patient with high titer would be the convenient patient to be included, maybe. And yes, but I like to reflect that, to thinking that in our experience, seeing a patient with higher titer and low titer, all of them reactive, clearly reactive, the reduction is the same during the first 12 months. So the curve of reduction is similar. So I talking about this topic because it's interesting your position, but maybe can exclude some patients possible to be included in a clinical trial showing a benefit. Even the titer is low, the reduction would be the same behavior in comparison with patients with higher titer. There is at least 3 papers or trial showing the significant reduction, even the patient remain reactive for definition, the reduction during the first 12 to 24 months after treatment. And personally, I think that the PCR, maybe as a strong evidence of effect during after treatment, the combination of serological tests showing a reduction would be an appropriate combination to show failure and success at the same time, using different ways.
So, Dr. Kirkhope, you think the variability in these serologic tests is so much that it's hard to sort of standardize, or can we say that to enter the trial, you have to be positive on, and then you specify the test, so then you monitor that same test as an assessment of response? Is that an option? So then you don't have these vagaries of so many different tests. Can that be done in a clinical trial setting, or that's not an option?
Yeah, I think you would certainly want to do that, but the question, like I said, we have very good serologic tests for detecting people pre-treatment or blood donors. But the problem I see is those targets may not be the best targets for being able to say something about cure before they get negative, or like as alleged for the lytic antibodies, but they would go negative faster. They may not be the right ones. And so, if just hypothetically, if we want to say, well, let's go with lytic antibodies. Let's say magically we could come up with enough native GP60 to supply that need. So we want to use that for follow-up, but I think we wouldn't want to use it for qualification because, the FDA hasn't looked at it. They don't know anything about it, and I would worry about false negatives and false positives using something like that that's not carefully tested. But I think that I was thinking as Sergio was talking, that, what are the barriers to doing this grand analysis? That, with all the different biomarkers and everything like that. And I say that the real one is the samples. Getting the samples and getting them organized and into a place where you can run a lot of them quickly. And then another barrier is getting the antigens, the recombinant proteins. But they're around, and some are even available commercially, and you could kind of do that. Maybe not all of them, and you could also get lysates and stuff like that. So I don't think that's a major problem. And and then beyond that, you just need money to have 3 or 4 people in the lab for 6 or 12 months or something to run this. But running the tests is not the problem. I mean, I've done hundreds of ELISA plates, and I've never used an automated system. And so, if someone bought me an automatic system, everything could have gone a lot faster. So I think the real thing is the samples, getting the samples. Good samples that haven't been frozen and thawed 50 times, and with the ink on the labels hasn't smeared, and there's more than 30 microliters and stuff like that. That's the real challenge.
Yeah, and then I think you have a comment as well? No, here's the thought.
We have to remember that serology is a surrogate of parasitemia. If we have a very good test of parasitemia, like PCR, we have to use PCR. If not, we are using something that is not that, and serology takes several years to demonstrate negativization. That was a standard for cure, but for acute patients. Never was set up for chronic patients. Nobody talked about that. This is a new information. But on the other hand, all the serological tests were set up for diagnosis, not for follow-up of the patients. Then we have several problems when we have to follow up patients during time. Then reinforce. We have a very good parasitological test that is PCR.
Yeah, I don't think anyone's proposing using serology and not PCR, or PCR and not serology. I think in the end, all of the above. More information is better, and I think the panel kind of agrees with that.
So speaking of PCR, here's a question for the panel. So I'm noticing quantitative PCR being used in the Chagas study, the E1224 trial, and then in the benefit trial. And those first 2 trials, although they may have failed, they actually seem to have very important data, particularly around Benzene. So the question is it the same assay being used in all 3 trials? And we've also been joined by Kathleen Whitaker from Center for Devices here at the FDA. And if she had any comments, I'd invite her to join in that discussion. So I guess the first question is it the same assay being used in all three trials?
Actually, the same assay was used in Chagas azole, E1224, and trial study. I think that in benefit, they started with another assay, and in the benefit they started with another assay, and then I think they changed to the validated assay because the study started before the validation effort we have done with the PCR. Yes.
The number of samples that you took, Isabella, you said that in one study you took one sample and the other three, or is the same number of blood samples for PCR?
Yes, that's an important difference. In the 1224 trial, we did 3 samples at each time point. The Molina trial, the Chagas azole, was one, and they did duplicate. We did 3 samples in triplicates. And I think that's part of the reason why they have 6% failure, and we have 20% failures identified, for example, in the benznidazole arm. And that might be related to the number of samples and number of triplicates. And so, and Stop Chagas actually used, it was Gentry's, and I believe they actually did 3 samples, right? They initially were going, Sergio can comment to that. Benefit did a single sample, because this again was before we had the results of the analysis and the evaluation that this was done.
In Stop Chagas is a single sample also for duplication the process, and there is a consensus up to date to use in all new trial. In fact, we are performing some clinical studies, use the validated combination of techniques, and it's very extended that the research team are accepting this protocol to perform PCR to assess treatment in clinical trial.
So that protocol may answer my next question, was if you had your life to live over again, how would you design a trial incorporating quantitative PCR? So it sounds like you all have thought that through and have some recommendations. Is that published for sponsors that they could access that?
We've been discussing that we need to publish this for a while. I think we have published the results, and there is a publication. There's an abstract for there's a communication to the European Congress of Pediatrics on the use of PCR as a marker of, there were 2, right? Go ahead.
We use 2 milliliters only for children. This is another point that we have to discuss. This is about the volume of blood. We are using 1 milliliter for newborns and 2 milliliters for children younger than 5 years, with very good sensitivity. No?
In the paper on the MSF trial that compares 5 versus 10 for adults and 1 versus 2 versus 3 mL, 1 versus 2 versus 3 samples, it should be published this year. So that will be coming out soon. Perhaps I wanted to go to the question that is on trial designs and endpoints. This issue on that one on the weakness and the strength of actually, and what is the evidence that change in serology a negative PCR or other lab test would actually be predictive of a later clinical outcome. I think that it's an enormous. This is actually the issue today, right? I think that from the Viotti data, you have the observational suggestion. We actually have the serological response being described, and in terms of seroconversion, and then you have actually the clinical outcomes. But no, that evaluation is not done. I think we're hoping that we're going to have the data from the benefit trials, that we will be able to see that, and perhaps from Triana. But I think that when you look at negative PCR and clinical outcome, I think one example that comes to mind for me is the one of immunocompromised patients. That's a clinical outcome, right? You actually have patients that have a positive exam, right, and then that are symptomatic, they have lesions, and then they respond. So it might be it's a somewhat different scenario because it's not chronic Chagas in that sense, but it is a relationship between that exam and a clinical response. Let's put it this way.
That's actually excellent.
Yeah, I think it's important to remember those, and also in acute Chagas, you could make those correlations in terms of symptomatology and so on. On the other hand, so those examples, I think, just to keep in mind. I think that serology, though, and reduction in titers and the prediction of clinical outcome, I think it should be said openly that also in the 2 placebo-controlled trials that were done, both in Andrade trial and the Sosestani trial, neither of them actually evaluated clinical benefit, right? These were trials that actually evaluated the response, because that was seen to be a surrogate of global burden of parasite in a way. But so unfortunately, no, for most of those we don't have today yet that the proof of later clinical outcome. I don't know, Sergio or the group, I think Karen wants to speak. Sorry.
Just related to Viotti, the data are scarce because very few people actually reverted to negative serology, but there was a significant association with reverting to negative serology and not having progression. I can show you the paragraph if you want to see. The P was 0.009, and the hazard ratio for those who remained seropositive in 3 tests over the course of the follow-up, the hazard ratio is 4.9 for progressing.
Do we know if the source data from the Viotti study exists still?
Yeah.
It does. Okay, that's good.
If I could just take a quick step back for John's question on, and a lot of these PCR assays we haven't necessarily seen, haven't come in, but one thing I just wanted everybody to keep in mind, the type of assay used and how, if you're looking at an FDA-approved assay, our assays are very different whether we're considering blood screening or diagnostic. They have completely different performance. They have completely different, the studies that we use to look at them. You know, obviously a blood screening is just going to be 99.9% negative people. And we actively go out when we look at a diagnostic for Chagas or whether be it serology or PCR, we actively look at chronic patients. We actively look at acute, we'll try and find acute patients. We divide them up into pregnant, non-pregnant. So, and we do find the results are going to be very different if you're going to utilize a quote-unquote blood screening test versus a diagnostic. It's a little thing that people don't necessarily think of, but it's surprising how different the results can be when you look at that.
So, Kiliana Starwajnik from Bayer. Returning to the question I had this morning before the previous session about loss to follow-up. I mean, the question is about reduction. So a patient that has been treated and you have observed reduction in serology levels and there is a loss to follow-up. And the question is this a patient of failure or not? And is there a way for us to predict that this patient will continue to, within the following months, to continue to observe curve? And this is a practical question, the thing that we discussed this morning, because we might not be able to follow these patients. Counting these patients as failure will be jeopardizing the results unduly because, there is a question, it's a possibility that it's getting better. So I would like to know what you think about and what the FDA think about it.
I don't know if I have a specific answer, but in general, we say minimize loss to follow-up because that's what we like. Because once you lose patients to follow-up, it certainly affects the interpretation of the results. And I agree with you, falsely classifying all of them as failures, you're really not doing anyone a favor. And I think there's a whole, I can ask Karen, but I think there's a whole publication on how to handle loss to follow-up. And I think the best advice is to minimize. I think it was the IOM, wasn't there a report not too long ago on missing data and how to handle it? So I think our advice always is to minimize it as best you can, and it looks like some people have been successful, so there may be some lessons learned from other trials that have been done, how this was done. And this is certainly an issue with, diseases of this nature because the kinds of people in whom you have to do the study or the areas of the world where these studies are done. And then typically we say in terms of the statistical analysis, it should all be laid out up front how you're going to handle missing data because we don't want you to post hoc then try to do different kinds of analyses and make the trial look better. So I don't think there is a straight answer to the question, but we're aware of the situation, minimize the loss to follow-up, but clearly specify up front how you plan to handle it. Is there anything else, Joe?
Just to add that an early time point for establishing treatment success or treatment failure will certainly help to minimize loss to follow-up. And I think that's partly why we're persevering on questions about PCR as an endpoint, because it would certainly help to minimize loss to follow-up.
Can I get back to a comment? Oh, sorry, you want to make a comment, and then I just had a question for Dr. Rivera.
Just to reinforce the comment that there are, in observational study, the correlation between the reduction of antibodies and the clinical benefit in observational studies in Argentina. At least 3 studies showing that. In addition, personally, I think that the PCR, the current protocol, is robust to show benefit after treatment as a qualitative result, real-time PCR as qualitative result, but there are some information, new information, like we are talking about reduction of antibody. The reduction of parasitic load is a new information also demonstrated during the first day after treatment and the effect during the follow-up. And it's necessary, thinking in this effect, it's necessary to remember that another information is that, for instance, the high parasitic load was demonstrated that it's a higher risk for congenital transmission. From mother with high parasitic load, the risk for congenital transmission is higher. So the effect of treatment, not necessary to become negative or absolutely cure, the effect of reduction of parasitic load traduces in a clear benefit avoiding congenital transmission for that reduction.
I just want to make sure I understood clearly. So on the presentation that Dr. Schickman made in your improvements in the PCR, you have factored in the variability between the different species, right? So that's not, hopefully not an issue with the current methodology that you're proposing.
Yes, we took into consideration. So it's actually, we have described it. We took them and we have they do have different limits of detection and quantification, but this is well characterized in terms of assay performance. And as one, and for the clinical trials, that was actually taken into consideration. Sorry. Perhaps I should just add one comment. It's something that we have done and we have not described. It's just a matter of detail here. It's just that we actually did genotyping of all baseline samples. We actually did RFLP. We also did sequencing of baseline samples, and we also did it for all treatment relapses. We also did that, actually trying to characterize the population and actually selection. So we did that. We actually did hemocultures in all the treatment failures also, and to actually looking for really a broader characterization there. And in terms of other assays, we actually did, besides the lytic serology, conventional serology, we did lytic antibodies. We did the ApoA1, also the multiplex diagnostic assay. And we have that for, for all time points. And we actually have those frozen samples, liquid. And they were all assayed at once in batch, because that's where the recommendations that we received. So at least I know that for some of the trials that we presented this morning, this is all available and could be reviewed. It will be a pleasure. And there will be samples also to be analyzed further.
Another point that is important, that doing PCR, the central point is the purification process. This is the critical point during the studies. We have to have a very good purification process. This is critical,
Back to the population, when you're looking at the PCR assays and you're validating, do we have a substantial population or cross-section of everybody, or have they been mainly validated in either pre-treatment or chronic? Oh, sorry. I was just wondering, when we're talking about accurate validation, which I firmly believe in these assays, when they have been validated, is it primarily in pre-treatment, or do we have a nice equivalent section of children, pre-treatment, chronic, acute?
So actually, the methods at the beginning must be analytically validated. So for analytical validation, we use artificial samples. We use seronegative samples that are spiked with known titers of parasites, and that's the way to know the analytical parameters of the analysis. So if the analysis has got a good analytical sensitivity, then we proceed with blind clinical samples. And in the first international study, we work especially with chronic Chagas disease patients, with samples from chronic Chagas disease patients, because they have lower parasitic loads, and was necessary to know the clinical sensitivity of the assays, and it was not more than 70% at that time. The best techniques gave, with one sample per patient, gave no more than 70% of sensitivity. And afterward, with the strategy we used for the E1224 study, using 3 samples per patient, we increased the clinical sensitivity to 90%.
Sorry, is it 3 samples being 3 separate blood draws, or I guess or 3 separate
yes,
PCRs?
there were three samples, independent samples, and then we performed the PCR by duplicate on each one of the samples, and if the sample was negative, we performed a third PCR from each DNA lysate, and with that we could enhance the sensitivity even 10% more. So if the patient had at least one PCR result was considered as positive and was included in the trial.
In adults.
Yeah, I'm
Because in
talking
children
only.
it's
Yeah,
a
of
little
course.
different.
Yeah, I'm talking only about this study because, as I mentioned before, these patients, this population has got very, very low parasitic loads.
I think perhaps to go back to the question also, if there is data across different populations and disease stages, and I think that basically there is quite a bit of data generated for sure before and after treatment in chronic Chagas, as indicated by Dr. Schiefman. There is also data in children using the same technique, different volumes. And there is data also in transplant patients. And I think there is data also from the oral transmission, from the foodborne using the same technique, my understanding. The issue actually with the oral is because it actually becomes so concentrated that there is a need for dilution, as I understand, right?
Yes, the patients, the samples we could analyze from the oral routes of transmission from the oral outbreaks had 8 logarithms of parasitic equivalents per ml. So we had to dilute it. It's the same when you analyze an HIV with very high viral loads. You have to dilute the sample and redone the test. Yeah.
But clearly this needs to be presented in such a way that this is all together, right, as a package. As I understand that as a device and treated as such. I understand the question from that angle. I think that there is. I think it's perhaps it's worth noting today that there is an effort from the Ministry of Health in Argentina and a funded project for the development of a kit, a PCR kit. The initial focus was for congenital Chagas, and there are actually now discussions on actually validating. It's actually in this kit for adult Chagas. So this is actually also under discussion, and it will be very good actually to have input in terms of if any additional, how to complement this information.
Yes, I'd like to reinforce the comment of Isabella saying that in the Minister of Health of Argentina, we are including at this moment the use of PCR in 3 specific situations. For acute phase in congenital infection during the first 3 months, if it's possible, the diagnosis. During access of treatment, and monitoring of reactivation in patients with immunocompromise.
Yeah, I mean, I think we'd certainly commend you for all the work you've done, and I think, as you rightly pointed out, it's the ability to look at all this information, get all the details ironed out before we can actually make a final decision. But we certainly congratulate you for all the work you've done over the years. It's great. Are there any other comments or questions from the panel members before I turn it over to Dr. Farley, because I think we're getting close to wrapping up this interesting day. Seeing no hands, I turn it over to you, John.
Great. Well, I know that a lot of you have traveled quite some distance to be here, and I want to assure you that it was worth it. This has been a very useful day for all of us. I think we in the office here particularly like these workshops because it's a rare opportunity to get all the right people in the same room at the same time actually talking to each other. And that tends to advance drug development. And we've heard loud and clear from our patients this morning that one of their big needs is in the United States is having drugs, having treatment options available and more readily available than they currently are. So I want to thank everyone. I want to remind you, if there was something you didn't get to say today, there is an open docket that the group that was made known to you this morning. That's available for your comments. I think this is the start of some dialogues that are going to continue. For drug developers, as you know, the division is available for meetings under the guidelines set forth under the Prescription Drug User Fee Act. And that happens very often, so they're just a reminder. And I want to wish everyone safe travels. And thank you very much for your time today. It was a great meeting.
And if I can just reiterate again that I'm hoping this is just not dialogue, that we come up with concrete plans to move forward in a rapid fashion so that we can get access to these drugs. Thank you.